Hello students, I'm Dr. Arpita De. A very warm welcome to my lecture today. The topic for today's discussion is phases of meiotic cell division. This is taken from the 3rd BSc Botany syllabus, paper 7, semester 6. Before we delve into the topic of meiosis, let's first talk about what is meiosis. It is a reduction division. This word is very important here. It is a reduction division where chromosome number gets reduced to half of its original number and leads to the formation of gametes. The entire story of meiosis pivots around this word reduction. Unlike mitosis, where we had seen equal number of chromosomes are being distributed to the daughter cells, here we see the number is getting reduced to half of its original number. That means 2n becomes n over here, and finally 2 sequential rounds of meiosis, that is meiosis 1 and meiosis 2, will be giving rise to 4 haploid daughter cells. Meiosis has got 2 sequential rounds, that is meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2 can be further divided into 4 phases: prophase, metaphase, anaphase, and finally telophase. Prophase 1 of meiosis 1 is a prolonged phase. Overall, S phase in meiosis is comparatively long in comparison to that of meiosis. Prophase 1 commences with leptotene. In leptotene, we see condensation of already replicated chromosomes. The replicated chromosomes begin to condense and are attached to the nuclear membrane via their telomeres. The sister chromatids become connected by meiosis-specific cohesion complexes known as REC-8p, and pairing of homologous chromosome is initiated in this phase. A string with beads appearance is observed where the beads are called chromomeres. Each sister chromatid is attached to the nuclear envelope, that is, ends of the chromosomes are drawn towards the nuclear membrane. Leptotene is followed by zygotene. Zygotene is characterized by the formation of synaptonemal complex between homologous chromosome. Synapses form of homologous chromosome is seen here, and it continues in the packetine stage which will follow. Synaptonemal complex is formed here. Tetrad is also formed. After homologous pair synapse, they are either called tetrads or bivalents. Next comes packetine. Here, crossing over of different chromatids occur. Crossing over of genetic material occurs between non-sister chromatids. Here, this is the main spice that add variation to the population. The paired chromosome become shorter. Once the synapse is formed, it is called a bivalent and crossing over can occur. The synaptonemal complex is complete, thus allowing chiasma to form. 2 sister chromatids separate from each other, but the homologous chromosomes remain attached. This stage is followed by diplotene. In diplotene, we see synaptonemal complex disappears and disjunction starts, but homologous pairs remain attached at the chiasmata. Finally, prophase 1 ends or terminates with diakinesis. The condensation of chromosome terminates here, the nucleolus disappears, and the nuclear membrane disintegrates prior to the onset of metaphase 1. Now let's just see an overview of meiosis 1. As the onset of meiosis, the DNA strands thicken into chromosomes. The homologous chromosomes, or the like ones, begin to approach each other. That means they come closer to one another. Once they come closer to one another, they form bivalents. The centrioles simultaneously divide and move towards the opposite poles of the cell. The bivalents duplicate to form tetrads or 4 chromatid groups. The nuclear membrane starts disintegrating, and finally way is made for crossing over to occur. In metaphase 1, the tetrads are attached to the spindle fibers at their centromeres, and they are aligned at the middle or the equatorial plane of the cell. This is followed by anaphase, where the tetrads get separated. That means what? They are pulled, and paired chromatids are pulled towards the 2 opposite poles of the cell. As anaphase comes towards its end and makes way for telophase, we see the chromatids have almost reached the spindle poles. The cell membrane begins to constrict. Finally, in telophase, what happens? The nuclear membranes enclose the separated chromatids. That means what? We see reappearance of nuclear membrane. The cell membrane completes its constriction. This is how karyokinesis gets over and makes way for cytokinesis. The first meiotic division ends. There are now 2 cells, each with the same number of chromatids as the parent cell. Now coming to meiosis 2. The prophase 2 begins immediately after the termination of meiosis 1. In the second meiotic division, homologous chromatids do not duplicate but merely separate. This is very important to keep in mind. They do not duplicate but merely separate. We should also keep in mind that it is not always meiosis 2 that will immediately follow prophase 1. It's a sequential cycle where meiosis 1 is followed by meiosis 2. In meiosis 2, in metaphase 2, the chromatids line up at the equatorial plane as seen in normal metaphases. The centrioles and asters are at the poles and the spindle is formed. In anaphase, the separated chromatids approach or move towards the 2 opposite poles, and simultaneous constriction of cell membrane starts to occur. Finally, we are moving towards the termination phase. Telophase 2 has been completed. Now what we see? 4 haploid cells are there, which contains exactly half the number of chromosomes which were there in the parent cells. This is how we complete the story of meiosis. Now, since we have studied meiosis, we should also know a little about its significance. Why did we study? The main important significance of meiosis is that it brings in diversity and variety within a population. How is it bringing that? It is by virtue of mixing of traits from the 2 parents. I hope you remember about the recombination that we were studying. This is how new traits are created. It maintains the fixed number of chromosomes in sexually reproducing organisms. Mutation takes place due to irregularities of meiotic division and repair of genetic defects in germline cells. These are the references from where I had downloaded the pictures solely for the purpose of imparting syllabus-based knowledge in this video lecture. So thank you so much for your kind attention to the lecture. Hope this video will help you. In case of any doubts and confusion, please feel free to get back to me in the comment section, and I will try my level best to clear your doubts. Thank you and see you soon.